and discuss few findings. Actually, I'm not going to talk about the findings, so I leave it to Gothamie Weerakoon, the PhD student of this project from Peradeniya University, to talk about the findings. So I'm going to give a brief introduction to lichens and why they can be used as nature's air quality sensors. So lichens as bioindicators. So you may have seen this title or the scientific publication, many scientific publications under this topic. But unfortunately, so although we have a very rich diversity within South Asia, especially Sri Lanka, being one of the world lichen biodiversity hotspot, we have never used them as air quality sensors. So what are lichens? It is, as we all know, it is one of the best symbiotic organisms in the nature. But I would say that they are semi-parasitic actually, or obligate association more than a symbiotic relationship. So to balance it, I would say it is a composite symbiotic association between a fungi, algae or cyanobacteria. Some are tricky, they have both algae and cyanobacteria, and additional bacteria, true bacteria are also there. So that is why it becomes a composite symbiotic association. It is not just one organism or two organisms or three, many. So I would call it actually a mini ecosystem that is capable of surviving without anybody's support. Just like you are sitting on a chair, it just needs a surface. It can be a human-made surface, it can be a natural surface. It just needs a surface to sit, but it is capable of sustainably living itself. So what is fungi? The main fungi is doing, fungi needs somebody to cook and make food. So it has invited or captured or farmed algae or cyanobacteria. So lichens are the world's oldest farmers. Before humans start farming, lichen knew how to farm cyanobacteria and algae and make their food. So algae and cyanobacteria, what do they get? They gain a substrate, they get a stable environment and mineral nutrients because fungi is a very hard organism. So more tolerant to environmental changes. So this is an electron microscope cross-section. We have used special coloring system to show you how the green algae round cells are embedded in a fungal high medium, actually. And the body of a lichen, we call it a thallus. Thallus, if you compare it to a flowering plant, so let's say a jackfruit plant. So jackfruit plant, they have roots, they have xylem and phloems, and protective bark, tissues and all. But lichens, they don't have any of these things. And thallus is unique because if you take the dominant fungal partner, or algae or cyanobacterial partner, they can be free living also, but when they come and combined or lichenized together, they don't look like the same. And they are one of the most slowest, the slowest growing organism in the whole world. So these are some of the recent findings. So it is not just one fungal partner, many basidio fungi yeast cells are there in the lichens. And this is a culture I made recently using lichen bacteria, so you can see that it is a true bacteria. When it comes to algae, these two algae are the most common in Sri Lankan lichens. There are about 150 different orders of algae within lichen symbiotic association, but these Streboxia and Frontifolia, this orange color algae, what you see usually on your coconut and king coconut trees, that is one of the most commonest algae in Sri Lankan lichens or tropical lichens. And this is cyanobacteria. These are the most common cyanobacterial genera that are found in Sri Lankan lichens, Nostoc, Rhizonema, and Stignomia. So this is one of our relagular lichens actually. So this photoband, Cypronema, is very critical because it can switch itself. Even it can slowly make food during the night. It really doesn't need sunlight. So it is a very weird cyanobacteria. So they come in different colors and forms. So if you look at them very closely, they are even beautiful than the corals. So I would call them, they are the terrestrial corals actually. And their forms and colors is due to the substrate and micro niche they live. And also the formation involves a large amount of unknown biology. We still don't know what is the role played by the second cells in our lichens. And most of the time that I told you that we have more than 100 different orders of true bacteria living in them, we still don't know their roles. Actually, lichens are very secret in their behavior, and they have given us a lot of space to study them. So for many generations, I might not be there to see them, but for many generations, many students, many researchers can study them. So these are some of the common growth forms. All of them are Sri Lankan lichens. This is in Europe. It is known as Christmas lichen, but even Christmas lichen is there in Singharaja forest, Morningside. So these are the most commonest growth forms in Sri Lanka. And out of all these growth forms, crustose ones are the most dominant in the country. And gelatinous lichens, compared to the other forms, they are very few and very hard to see, especially during the dry seasons. Always they are overlooked as algae or green algae or brown algae, but they are truly lichens. So this is one of the very well-known lichen. People used to call it old man's beard, but I will tell you why we are not calling it old man's beard anymore. And I'm always questioned, how old are lichens? Lichens are older than dinosaurs. So lichens appeared on the barren terrestrial Earth more than 600 million years ago. So they are capable of starting succession in any barren environment. And these are some of the amber fossils and also some rock fossils that have been discovered to show them that they are older than dinosaurs. And I told you, they are the slowest growing organism in the whole world. This particular frigid ice cold button lichen in Antarctica, the growth rate of this lichen per year is 0.036 millimeters. And that is why they have become very good sensors, actually. So they grow very slowly over the time, hundreds of years, and this particular lichen that is shown on the photograph is 11,000 years old. So they say us what has happened over the climate change, how the temperature has changed or increased or decreased, and how much humidity was there, and so on. So we can use lichens not only for measuring air quality and also evaluating habitat quality, but we can use them for to date past events and also to determine age, especially the lichens that are growing on monuments, like in Anuradhapura and Polonnaruwa. If they have retained, so you can get to know more hidden data, more hidden stories about them. And they grow on any surface, even man-made surfaces as well. And if you observed very closely, I'm pretty sure each and every single garden, home garden in Sri Lanka, you have more than 100 lichens. So I guarantee that over 100 lichens within your own home garden Only thing is that we are never looking at them. We don't care. They're just there, sitting there. And these lichens who are just sitting in your backyard or your home garden, they are secondary compounds, so their chemistry is very important. They are important in pharmaceuticals, cosmetic and food industry, so that that the use of lichens and their worth in cosmetic industry in Europe is over $600 billion per year. And more than 1,500 metric tons of lichens are collected from Nepal and India each year and exported to Europe. It is not a good practice, but that much lichens are used in industry. And for thousands of years, many people have used them as spices, herbal teas, and dyes. And some of the very expensive clothing, garments like tweed, that gentleman's tweed material, they are completely dyed with lichen colors. So they are capable of making their own sun cream, that most of the sunblocks and creams that we use in the tropical countries, especially in the hot sunlight, so that compounds are actually collected and used from the lichens. So many of the desert lichens that have tons of these secondary compounds, they are very, very colorful. And this is something that few examples how insects and animals, they use lichens for mimicry, and very close relationships like this. For their own protection, sometimes they use lichens, and also as food they use lichens. So how many lichens do we know up to so far? Less than 20,000. So out of the 1.5 million fungi that are in the world, so only 20,000, less than 20,000 are lichenized fungi. So how much is known about lichens? So you see, so we have been studying lichens in Sri Lanka for more than 150 years, but still Sri Lanka is within this lowest studied group of the world. And these are some of the places that foreigners or foreign researchers have collected lichens from Sri Lanka. The very first lichen collection was made in 1863 by a very famous botanist called Thwaites, who was a botanical garden director during the English colonial time, and rest of the collection made by other foreigners. Unfortunately, they are in foreign herbaria. We Sri Lankans don't have access to them, but recent collections made since year 2000, we have now deposited over 3,500 specimens, fully identified specimens in the National Herbarium in Peradeniya Botanical Garden. So if you want to study any lichens, want to compare your collection and identify them, so that is the reference collection that we have in Sri Lanka. So since I started studying lichen in 2009, April onwards, so I focus on these objectives, especially to identify the lichens that can be used as air quality indicators. And Sri Lankan lichens, they are habitat specific. They like primary forest or undisturbed habitats. Their richness increases with the habitat quality, and they like bit of canopy cover. They don't like to be exposed, fully exposed to sunlight, and also variety or the diversity of trees in a habitat. It is very important for lichens to survive and thrive. So these are some of the forest surfaces that we use. So this is a tag tree from the Rilagala forest where we have our permanent monitoring plot. So this tree was identified and tagged by our team members, and we have been studying all these lichens on the tree trunks for our study. But on the other hand, Sri Lankan diversity on these wet rainforest leaves Nobody has ever focused on it. So there could be over 2,000 different folliculus, or the leaf living lichens could be there in Sri Lanka. Unfortunately, not more than 150 species been identified. So that is a sector that Sri Lankan students and researchers can focus on. And current lichen checklist, when I started as a PhD student on the project, so only 463 lichens were known to Sri Lanka. Today it is almost 1,500, so we added about 1,000 species within last 15 years. And out of that 1,000 species, over 100 are new species that we described. They are new species to the entire world. And 900 new records, or over 1,000 records, and more than 200 are new to Indian subcontinent or to the South Asia, and over 30 are new paleotropical or all world records. And to discover new lichens, that is one of my hobby, to identify new species. So these are few of our new species, and you can see that how colorful and beautiful they are if you look at them very closely. And we have an online key that we prepared. Over 200 lichens are there. You can freely download it to any smartphone. Using this key, you can identify most of your backyard lichens and also montane. Like if you go to Hantana or Haggala or Horton Plains, this key, if you take it on your phone, you can get to know the names of the lichens that you are seeing next to you. And these are some of the things we have been doing during last 10 years. We collected fresh samples and we analyzed them, especially their DNA data, to place them correctly in the correct families and genera, because some of our lichens were named by foreign lichenologists, and they gave European or Northern American names to our lichens, which are not actually true or correct. So correcting the 463 lichens that were documented until year 2000, so we did a lot of corrections. So Usnea barbata, this is old man's beard lichen. For over 150 years, this was known as Usnea barbata or old man's beard, but actually we don't have that lichen in Sri Lanka. Or we don't even have this lichen in India. So the reason was that this lichen was first collected by one of the European researchers, and he simply named it as Usnea barbata or old man's beard, following the European lichen. So even our Usnea barbata is not in that correct genus. It's something called completely different genus called Ramalina. So we have been doing. So my pet family is family Graphidaceae. This is the largest tropical lichen family. And we know over 2,500 species in this particular tropical family. And Sri Lanka is the world hotspot for this family. So we have nearly 520 species, and out of the new species we described, majority is under this family. This is the most dominant family in the tropics, and we have the highest number of new species discovered during the last 10 years from the whole world. And in that family, this is my two favorite genera. We call them strip lichens. So when it comes to this particular genera, Graphis analographa, so out of the newly described species, highest number are described from these strip lichens. They look the same, but if you take them under the microscope and cut them with a simple razor blade, you see that how much diverse they are. And I made an attempt to understand what could be the total richness, or how many lichens could be there in Sri Lanka. So I used this forest map and grid that made 10 by 10 square kilometer grids, and entered the number of lichens I know for each and every single grid. Unfortunately, that some of the northern part of the country and also east coast like Batticaloa and Trincomalee, I have not done much work. Horticolous lichens that I study, but however, with the known that the green parts are the parts that I have surveyed more during the last 10 years. Red sites are still, there are so many places that we have not surveyed. So, but still, with the data that is available and in hand, I can say that tropical rainforest, that where Singaraja is, the highest number of unknown species can be discovered from those areas, 226. That is my prediction number, that to be discovered within each 10 square kilometers. And then when it comes to Horton Plains and Haggala area, and also other mountain areas, we have done quite a lot of work, but still more than 200 species can be listed from each 10 square kilometer plot. So I would say, so when it comes to the lichens that are growing on tree trunks, I'm only 5 feet tall, so I can't see much unless I use a ladder or climb the tree. But even within my height, I'm saying over 600 species are still waiting to be discovered. So if you are 5 feet 5, you can imagine it will be 1,000 species. If you are 6 feet, it will be 2,000 species. So more taller, it is much better for lichen studies. Unfortunately, I'm 5 feet tall. Okay, so lichens as bioindicators. So this is the most interesting part out of everything I do for me as an individual. And I thought, okay, SNH project is a good opportunity, good platform to test this thing, which I like. So we joined our hands. We joined our hands with Dilmah and that this is my 10th year that I'm working with Dilmah. I started working with Dilmah or collaborating with Dilmah from 2014. And 2015, we could publish a simple guidebook for Sri Lankan lichens. So my journey with Dilmah, it is 10 years now, so that I asked SNH colleague and Professor Nisankar also. So we joined with Dilmah from 2019, and we prepared we demarcated our plot in Rilagala Forest, where we know a lot of lichens from Queensberry Estate. And we have even discovered several new species and named one after Queensberry Estate. So these are some of our findings from there. So we identified over 130 species itself from a 40 by 60 meter plot, or 40 by 20, Buddhika. Okay, so he will give you the exact length and width of the plot. So out of that 130 species we identified within our permanent plot, 60 species can be, it means like 50% of the species known from the plot, or almost 50%, can be used as significant indicators. And then, okay, I know some lichens from Knuckles, I know some lichens from Horton Plains, I know some lichens from Haggala Botanical Garden, and also Singharaja Forest, and also Vilpattu. I have done a little bit of work in Vilpattu as well. So I thought, why not I throw them all on this DCA ordination and see how many lichens can be effectively used in Sri Lanka, a small island like ours, as significant indicators. So this is what I got. So not only we have very rich diversity in the country, but also our lichens can be effectively used as air quality sensors. So these photographs are all from Gothamie Weerakoon, his PhD study. You can measure the lichen response to, especially air pollution, with their growth, whether they die or live due to critical levels, whether they will detach from their surface and fall down, and how the color will change. This is more like coral bleaching. Corals, they lose their color because of El Nino and other effects. Same thing, when pollutants are there, when the temperature goes high, the lichens also bleach, and also they become aging, or they become inactive and very old. So this is a very good example. This is a healthy thallus. That is what is happening with the ammonia. So my colleagues, Professor Nisanka and Mark Sutton, they talk about it, and Ajinkya, he told you how to play with the climate numbers. So this is what we see after the air pollution goes up or surpasses the critical levels. And Buddhika, he did the very first nitrogen transplant experiment that was ever done in a tropical country. This is a very novel study in whole world. He used 2 different lichens that are well known in our site, and he took sampling, or he measured them 6-time intervals of exposure to high level of air pollutants, and he measured electrical conductivity, chlorophyll content, carbon-nitrogen ratios. All these measurements were done in Edinburgh University. So we know what is happening with the lichens when they are exposed to high amount of air pollutants. And it is very promising what we see from this study, although it is a novel design for the entire world. So that is why that we can guarantee we don't need high-tech, very expensive air quality monitoring equipment. That is very expensive, millions and billions of dollars to invest to identify the problems we have due to habitat destruction and degradation. We can use the lichens as very high quality indicators. So this is another technique used by Buddhika Weerakoon. So he used some time-lapse cameras, and he took images from cyanobacterial and algal lichens each and every single day at a particular time, whether it rains or dry. The camera, when taking the photographs, and this is what we see. So this is how the lichens look in 2022. This is what happened after 1 year. So you can see, even without high-tech equipment, within a short period of time, lichens can show these changes. They can respond to the habitat changes and air quality changes very rapidly, and they are very effective in the nature. And some of the other students, especially from Kelaniya University, they have worked on secondary compounds or bioactive natural products of lichens. They selected lichens in very harsh condition or hard environment in Sri Lanka, mangrove ecosystem. These are some of their publications. So you can see lichens are not only effective as air quality sensors, but they have lots of novel compounds that can be used in pharmaceutical industry to develop or bring dollars to Sri Lanka. I would say bring dollars to Sri Lanka. So these are some of their publications. Okay, the very last slide. What is happening to Sri Lankan lichens? Okay, Sri Lanka is a biodiversity hotspot. The family Graphidaceae, the highest number of tropical lichens. We are the hotspot, global hotspot in the whole world. We have discovered highest number of new species per 1 square kilometer in whole of the world. So, but what do we know? Them. Are we taking necessary action to safeguard this resource? Because most of the new species we described, they are point endemics. They are only seen in 1 or 2 places in Sri Lanka. Only in Horton Plains, or only in Knuckles, or only in Singaraja. So what have we done enough for them? Or do we know enough of them? Unfortunately, no. So Sri Lanka has become the country that recorded highest number of new species and new records per unit area in the region and also for the whole world, but we are not doing our, we are not paying our due respect to this valuable component in our biodiversity. So with that take-home message, I like to end this presentation for this evening, but you are most welcome to contact me or contact us, actually. We are hoping that we will be able to continue our work for another 3 years in Sri Lanka, and UK government will generously fund us again. So let's keep our fingers crossed and hope for the best. Especially, we want to ask this audience to take this message to whoever is responsible and whoever can help to secure this very important biodiverse component. Thank you.